In this seminar, I will be introducing the techniques of variance partitioning and commonality analysis. Both terms mean the same thing and can essentially be used interchangeably as they have the same underlying methods. Their purpose is to provide a way of understanding the amount of unique and shared variance explained by explanatory variables used in redundancy analysis, canonical correspondence analysis, and also for the univariate case of multiple linear regression. And these variances tend to be visualized in a Venn diagram format like on this slide here, where you can see the unique variance explained by the yellow variable, but also where it overlaps with the red and blue variables. And this is a pretty exciting follow-up technique to these different analyses that not a lot of people use, but can actually help pick apart some of the issues that you kind of run into with techniques that are based on regression. So the reason this is exciting, we can illustrate here quite easily with this ordination plot resulting from a redundancy analysis. Looking at these plots, I've only labeled variable 1 and 2, and you can see they kind of point in the same direction and the same magnitude. And this makes it kind of hard to differentiate between the effects of these vectors on our response variables, and which ones have stronger effects, which ones have weaker effects, etc. And how much do they overlap and how much variance they're actually explaining in our response variables. You can see this even clearer if we plot these two variables against one another. You can see there is in general a slight linear relationship between the two of them. Because these variables are related, they'll behave similarly if we use both of them in a model, which is an issue that we call multicollinearity in regression. But looking at this plot, you can see they aren't perfectly, linear. So we know that there is some unique variance that they will explain, but we really can't quantify how much they are actually doing in our model. And that is the genius of this variance partitioning method. It actually gives us a way to quantify the unique variance explained by it, but also the overlap, which is the effects of multicollinearity we see in these different variables. And a way to think of that is with these Venn diagrams, right? So you have the unique effects of variable 1, yellow, and the unique effects of variable 2, or variance explained by variable 2 in red. So you can see there is variance uniquely explained by both of these variables, but there is also overlap. And with this method, we can quantify the parts of this different Venn diagram to see how much overlap there is compared to how much unique effects are explained by each of the variables. So just to give you an idea of some scenarios where these methods would be highly relevant and useful, there's this kind of foundation of these methods within environmental science. Is this example in community ecology where you have a spatial gradient at your sites, and you also have some measured environmental variables in your study, but you're not sure how much the results of the species abundances you measured were actually due to environmental variables that you measured at your different sites or due to some underlying spatial gradients that you did not measure. So before all you could say is there's some confounding results in our evidence, there's some covariation at the plots that we're not exactly sure. We can say this is happening, but we can't actually quantify how much this is affecting species abundances versus how much our environmental variables we measured are actually affecting species abundances. So the real benefit of this is it allows you to quantify these two things as well as the overlap between these two variables. Also, an example for the univariate case, you might be trying to examine how multiple climate variables affect a single response variable. But these climatic variables are inherently related, and this is useful for any linear regression where you have explanatory variables that are likely related, and it's difficult to see how much variation in the model is explained by the individual variables, whether these variables are overlapping because of multicollinearity, they have unique variance that isn't explained by other variables. It's hard to say this without this type of method. And really what it does is it helps you understand how your different response variables behave with one another in your model and what their unique effects are, as well as what their shared effects on contributing to variation explained by your different models. So now I will explain how variance partitioning works for redundancy analysis and canonical correspondence analysis, and then we will work through an example of this together, because I think it makes a lot more sense if you can see how these fractions and whatnot actually look when you're calculating them. So the high-level explanation of this is that we can calculate the variance explained by our full model containing each variable of interest, and then we can extract the partial variation of each variable by creating partial models and extracting variance explained by these to calculate variance explained by each component of our full model. So in the way we do this for the multivariate case, we assume the variation in our data equals 1, and the variance explained by the full model is given by the sum of the variance explained by the axes. And we already know how to calculate this. For RDA and CCA, it is the eigenvalue all over the number of variables in the model. So we calculate the variance explained by the full model first, and then we create partial models, partial RDA or CCA models containing only one or a subset of variables. And we calculate the fraction of the variance accounted for in these partial models. And then we can subtract these fractions from the full model to figure out the variance explained by each part of the Venn diagram, if we want to think about it that way. But I think it will become much more clear what this actually means after we work through an example. So to help illustrate this better, I'm going to keep the Venn diagram here so you can see the different fractions that we are calculating. So when we calculate the variance for our full model, the model containing all of the variables, we are calculating fraction A, B, and C, which are labeled here in the Venn diagram. And the code for how you do this is shown right here. You do an RDA, and then you extract the R squared. And the reason we use R squared in this case is because some of these variables might have, different, especially if you're doing an RDA, these might be tables of variables, so you might have different numbers of variables in these tables, which will cause your RDA, your R to be inflated because R increases with each variable you add to a model. So we actually want to adjust this R squared for the number of variables in our model so it's not inflated and therefore not comparable between models. So the first step, we do this full model calculation and we get this value. Then we'll save the value here while we do our second step, which is to calculate fractions explained by each variable or set of variables by creating individual models for each variable. In this case, we only have to do that 2 times because we only have 2 variables in our full model. So when we calculate the variance explained by variable 1, we are calculating all of this yellow circle here. And when we do it for variable 2, we're calculating all of this circle here. So it's BC for variable 2 and AB for variable 1. And that's this value here, represents fraction AB. So now we have all the values that we need to do some basic algebra to be able to extract the individual variance of fraction A, B, and C so they're not combined in this ABC, AB, or BC. So first we calculate B by adding A, B, and BC together. So we add this circle to this circle. And what you notice is we have 2 B's if we add that twice. So we can extract A, B, C, and only one A and one C. So if we extract the variation explained by A, B, C, we are left with only B, which is great because then we can go on to use this to subtract from our A and B fractions to be able to calculate each variable individually. And then you can go on and display this in this Venn diagram here very nicely. Everyone can be able to understand the variation, the nice way to visualize the variation explained by each component. That's also a good practice to calculate your residual as well, which, as we mentioned earlier, is assumed to be 1 minus the variation in the full model. I'm not going to show the values here because you will walk through this in the lab and I don't want to give you the entire answers, but you will be able to calculate this yourself as well. So that wraps up the example of the multivariate case. And I wanted to show you how variance partitioning with the univariate case works, but extend this to 4 variables, which is kind of the maximum recommended allowance with variance partitioning, which you'll maybe understand at the end of this slide. So the only really difference with univariate and multivariate is you're working with multiple linear regression rather than redundancy analysis or CCA. So what you actually end up using as your variance explained for each model is r squared for the multiple linear model instead of the sum of the eigenvalues all over the number of variables in each axis. So for this, let's say we want to figure out what the unique contribution of variable 1 is. In that case, that ends up being quite simple. It's just the r squared of the full model minus the r squared of a partial model containing only the last 4 variables, excluding variable 1. And we can see why this is the case by looking at a Venn diagram. If we look at all the areas that aren't overlapping with variable 1, we see that, or all the areas that are overlapping with variable 1, these other variables, if we create a model with all of them, what remains is variable 1. So it's simple to calculate the unique contribution of each variable, but let's say we want to calculate the overlap of all 4 variables, A, B, C, and D. Think for a second how you would go about doing this. The issue, right, is it's not as simple as, well, it was just 2 variables and we could just add the whole thing up. We could create a model for each variable and add them all together, because if we do that, we end up getting an overlap 4 times of A, B, C, and D, which makes this quite complicated to get rid of this overlap 4 times. If you want to try your hand at figuring out the algebra to solve this, you are welcome to do so. However, there is a method in R in the vegan package that will just calculate it for you, and you can output these nice Venn diagrams. You don't have to make them by hand, which I would recommend in doing in all cases. We just walk through the methodology for the 2 variable case because 1 is easy to understand with 2 variables. The calculations are quite simple, but anything above 2 variables, I would recommend using this method, as it makes your life quite a bit easier and it makes the plots for you. So before I let you all go on the lab, I just want to make a few last remarks on this method. So this is mainly a tool for understanding the relationship of your response variables with one another and how they fit in your model. Understanding relationships that you don't get just from the output of an RDA, CCA, or a multiple linear regression. It's more to enhance your understanding of the model rather than to be used in a model building or selection process. So that leads us to point number 2, that high amounts of variance does not equal importance in the model. This does not make one variable better or worse than one another. It's just telling us how the variables relate to one another, and it enables us to draw new and interesting conclusions from that. So yeah, so this is a danger with this. If you are only interested in a predictive model, this isn't actually too big of an issue, but if you are interested in trying to say how these variables explain your response variable, as with any regression-based analysis, you do need some sort of theoretical background, because otherwise these could just be casual relationships that you are seeing. I also wanted to make a note that you might sometimes see this message here where it says values less than 0 are not shown. So that means you are having negative variance partition show up. And what this tells you is this variable 4 actually is inhibiting the effects of some of your other variables that you are seeing, which also tells you this might mean that variable 4 has like an opposing slope and is in some way canceling out the variation explained by some of your variables, which is good to know. So here are the references I used in constructing this presentation. I will also note anything that you see written by Burkhardt, Legendre, or Drapeau. They all really contribute a lot to our understanding of variance partitioning within the field of ecology or community ecology. So if you see any papers by them, if you want to look more into variance partitioning, they all have been hard at work disseminating this information to the larger science community. And with that, I hope you enjoy the lab and look forward to any questions that you might have in our meeting for this seminar.